Welcome to the third week of our summer series of the Wednesday evenings at the Genome. Great to see everyone here again this week. And a good turnout on Zoom as well. I'm really excited to introduce Dr. Sumi Jayadev, who is one of really the pioneers in Alzheimer's disease and really leading the field both here on the local level as well as at the national level. And I'm really excited to hear her talk today. For those of you who do not know Sumi, she is a professor in the Department of Neurology here. She is the Vice Chair of Research for Neurology. She also is in the department or the Division of Medical Genetics, where my appointment is. She's the Associate Director for the UW Alzheimer's Disease Research Center, where she's also the clinical core leader of the UW ADRC, which is Alzheimer's Disease Research Center. She does a lot of really awesome work in terms of both the molecular basis of Alzheimer's disease, as well as working with the patients directly. She's a clinically trained neurologist and helps and runs actually the neurogenetics clinic here at the University of Washington, where she works directly with individuals who have both early onset as well as later onset Alzheimer's disease, as well as a whole host of other neurological conditions. She is, phenomenal reviews both on the clinical side as well as the research side, as well as she's just a great teacher and great person to work with. So I'm really excited to hear about the phenomenal research she's doing today. So thank you very much.
Thank you so much, Andrew. That was extremely kind. So I am very glad to be here, and thank you for the invitation. So we're going to be talking about how do immune cells drive Alzheimer's disease. But first, I'm going to give you a little bit of a roadmap. Three major sections. We'll talk about how human variation contributes to the risk for Alzheimer's disease. How does that variation alter the function of immune cells? And then I'm going to give you just an example of how immune cells can go awry to contribute to Alzheimer's disease. But first, before we answer that question, I want to go a bit way back and say, why even ask about immune cells in a brain disease? Isn't that just made up of neurons? And so here is going to be the star of our talk later on today, microglia. That's one of the brain immune cells. But I'm going to take us back to the history of Alzheimer's disease. So Dr. Alzheimer, who is right here, had been taking notes about his patients, the clinical features of his patients, the neuropathological features of their brain tissue, and had noted these interesting aggregation of proteins or aggregates in between the neurons, so out in the extracellular space, and also that there were aggregates even within the neurons, so in their body, in their processes. And those two features really became sort of the pathognomonic definitive diagnosis of Alzheimer's disease. Even today, when we suspect someone of having Alzheimer's disease, we look at the brain autopsy, and if the brain autopsy doesn't show amyloid beta and tangles, we think, well, maybe there's something else going on. So since that, more than 100 years ago, when we first appreciated plaques and tangles, we've made a number of important findings. And two of them have been particularly influential in the way Alzheimer's disease research has progressed over the decades. So the first one was in 1984, where we had finally figured out what exactly is in that amyloid plaque, because amyloid just means the configuration of the protein or the peptide. It doesn't tell you what that protein is. So investigators were able to sequence the amino acid sequence of that peptide and found that it was from a protein called APP, and the peptide is called amyloid beta. And the next piece of information came in the 1990s. There were investigators who've been following families that had early onset Alzheimer's disease, autosomal dominant Alzheimer's disease. So there were families with generations and generations, and we knew that there were important biological clues in those families. We just had to get to them. And in fact, Dr. Tom Bird, who is here from the UW, he and his team had identified one of those genes, presenilin 2. So over the 90s, we found these three genes, presenilin 1, presenilin 2, and APP. And those were all sufficient to cause Alzheimer's disease. Just a single mutation in that one gene could cause AD, which of course told us that that was really important. What was amazing is that we found out that those three proteins, the proteins that are encoded by those genes, actually interact. And so APP, one of the Alzheimer's disease genes, encodes a protein that sits in the membrane. And then presenilin 1 and presenilin 2 encode proteins that actually are also in the membrane, but they have an enzymatic property, and they cleave APP to release amyloid beta, the peptide that's in the plaque. So it's incredible. These three things are biochemically related. It really seemed like we have this nailed down. And so that led to what we call the amyloid hypothesis. And you may or may not have heard that when people talk about Alzheimer's disease. And here's the principle, that we know that mutations in APP and presenilin lead to amyloid beta aggregation. We know that in the brain, there's deposited amyloid beta peptide. We imagine that that is associated with the disease itself. And so there was enough information that those amyloid proteins could be toxic to cells. And so the hypothesis is that this amyloid beta leads to stress in the neurons, and stressed neurons start to develop these tau tangles. That was the other piece of pathology. They develop the tau tangles, which eventually leads to neuronal dysfunction and death and dementia. So given that, it seems like the obvious intervention really should be first, let's just get rid of the amyloid if that's what's causing these downstream problems. So we began to test the amyloid hypothesis. And one of the great things about having this genetic form of Alzheimer's disease is now you had a tool. So now you could take a mutation, a gene that you know causes disease, and then you can put it into an animal, which allows you a number of benefits. For one, you can see how the disease progresses over the course of an animal's life. So you can see if the degeneration changes, if there's cell loss, if there's any type of reaction. You can also use these models to screen compounds. So they've been the workhorse for neurological disease. And by the late 1990s, we were able to test that hypothesis in mice. And so investigators had taken a mouse, and just to let you know, when we make mouse models of Alzheimer's disease, we can't just put one gene mutation in there. You actually have to put 3 mutations, 3 different mutated genes into the mouse in order to develop the same pathology that humans do. So that's one clue that there's a difference between mice and humans. But anyway, so this first study, they were able to vaccinate mice against the amyloid peptide, and that mouse cleared its amyloid. So that was terrific. We're able to intervene and remove the amyloid. And so since then, we've had a number of decades of trying to do the same things in humans. And so we started out with an active vaccination, and that didn't work out. People had trouble with that. And we've been doing passive immunity, so giving people antibodies against amyloid beta, and there have been a number of failures. And so we initially would get failure after failure in terms of removing the amyloid. We also tried to inhibit the enzyme that creates the amyloid, and that didn't work out. So after a number of tries, there may be a few compounds that might show promise. And so that's a great step forward in the sense that we can feel more confident that we are able to attack a biomarker of the disease. And the FDA just approved a second one. But I think there's a number of us in the field who feel that we can do better. And there's still more to be learned from our patients. And so what are the challenges and opportunities in Alzheimer's disease? Because they are challenges, but we can turn them around and leverage them to learn more. And so this is, it's a little dramatic of an image, but this is the idea of the translational valley of death. We have scientists on one side. They do a fantastic job of understanding the biology of a cell in a dish or animals, and then they produce candidate molecules, candidate medicines to try. The problem is we can never seem to surmount this huge chasm, and we have a real difficult time getting drugs into the clinic. And this is particularly a problem for neurological disease as well. We have a really poor hit rate. So why is that? Well, there are a number of things. So first of all, people and Alzheimer's disease pathways are really diverse, genetically diverse. So it's not one size fits all. We're going to need to be more specific when we think about how to intervene with disease. And second, I alluded to this a bit, these animal models, while they've been extremely helpful, have their limitations. And in the end, The animal genome is different than the human genome. And when we're talking about these complicated cell-to-cell communications as a way of, as mechanisms of disease progression or disease resilience, then it really matters what animal you're talking about. And then lastly, we've learned in some part through the trials that we've been doing that we really need to start treatment early. And I'll explain a little more about that. The talk today, though, is really about this first point, that people in Alzheimer's disease pathways are genetically diverse. I will make a couple of points, though, about the disease models. There are other advances that we're doing where we can study these processes in the human genome, and that there are efforts to improve our biomarkers or to develop new biomarkers. So with regards to the models, I mentioned that we would like to use a model that has the human genome. And so there are ways that we can do that, one of which is to create stem cells. And so we take a sample of skin or blood or some sample from a patient. We can reprogram that into a pluripotent stem cell, and it's pluripotent, so it can develop into different types of cells. And then we can take that stem cell and differentiate it into the different types of brain cells that we want. And then you can culture them and do experiments in the dish. And this is really important for a number of reasons. Not only is it in the human genome, but it's also in a dynamic system. So now you're not just observing changes in disease tissue, you can actually create hypotheses, you can test them, you can see how different compounds work. And that's really an irreplaceable type of disease model. Here's just a immunofluorescence image of what that looks like. This is a microglia, an immune cell in green, and this red is a neuron, another brain cell. And then there's also interest in trying to make more complex models. So these are three-dimensional models, brain organoids or assembloids. They're just multicellular aggregations of cells that actually can form their own organization. They self-organize, and they can be really valuable to study more complicated cell-to-cell communication. So I mentioned the biomarkers. So on this graph, on the x-axis is cognition. So on the left is cognitively normal, and on the right is demented. And then the y-axis is the biomarker, so from mild to severe biomarker. And what we knew from earlier studies is that when we were starting to do trials, when we were using these antibody trials, we were, for the most part, really working in this domain. We were identifying people that had Alzheimer's disease or that had cognitive impairment, and then we were giving them drug. And the problem is, by that time, the horse is likely out of the barn to some extent. And so how do we get there earlier? And, and there can be different ways that people can be high risk or low risk, and high risk might have even earlier onset. And so there's some variability in the way the disease actually presents itself. But particularly with the familial forms of Alzheimer's disease, we've been able to learn a lot about the biomarkers associated with AD. And what that means is these are changes in the system that you can measure even prior to clinical obvious cognitive change. And so these are a number of different biomarkers. So in red is an immune biomarker, and I'll talk more about that as far as what that biomarker actually is. And then there are these other biomarkers that you may have heard of. So amyloid PET is a PET scan where you can, where we look for amyloid beta deposits within the brain in the living person. So as an alternative to autopsy. And that's been one way we've been able to screen for people who seem to be having Alzheimer's disease pathology, but haven't yet developed any clinical symptoms. And then we can also measure other things, say, in the CSF. So we take a lumbar puncture, we get this fluid that's actually surrounding the brain. And those molecules also change prior to clinical involvement. And then there are some other metabolism changes. So there are ways that we can find folks earlier. We're still working on it, but those are some of the things that, that have been a challenge that we're getting better at. So human variation. You know, we clearly have a lot of genetic variation. Some of us have black hair, brown hair, and that same sort of variation can influence how we are vulnerable to disease and which diseases, et cetera. And we can try to leverage that information to start to hone in on the biology of disease. So what I'm going to show you is I'm going to be talking about polymorphisms, which means changes in the gene, in the DNA structure, that they aren't rare mutations. They're just differences where one person has one version and the other person has another version. So if we look here, there's a cell and we have a piece of DNA and we zoom in on the DNA. And then here is, these are nucleotides. These are the building blocks of DNA. And you can see there's some sequence A, T, G, C. And so one version of this block of DNA may have a C in this position, a nucleotide. And then there can be another version that has an A in this position. And that's what we're calling a SNP, a single nucleotide polymorphism. And so we can say there's a version 1, a version 2 of these, of this block of DNA. And then we can also measure that in the population. So for instance, this version is 80% of the population, this version is 20% of the population. And we can now use this variation to start to get more specific about where in the DNA we're having problems. So a very powerful method to understand genetic variation across the entire genome is our genome-wide association studies. And so we've had these for a number of years, and I'd say in part because we weren't ever going to be able to, at the time, we weren't able to directly sequence each nucleotide base by base by base. What we were able to do is find these SNPs that tag a chunk of DNA and then try and make an association of that chunk of DNA with the presence or absence of disease. And that's what a genome-wide, so it's all the genome association study, which parts of the genome are associated with AD. And so here's a Manhattan plot. I think many in this audience knows this. A Manhattan plot, and what these are is each little dot is a SNP, and this is across all of our chromosomes. And it's measuring the association of that SNP with the presence of Alzheimer's disease. Is it statistically more present in Alzheimer's disease compared to control? And there is a way that we can test the power, the significance of that association. And the really high dots here are most significant. And so what we find are a number of regions all throughout the genome that seem to be more present in people with Alzheimer's disease compared to control. And remember, these SNPs are not just marking one nucleotide, they're marking a chunk of DNA. So in these chunks of DNA, there reside different genes. And so that's what these names are. The genes that are in that chunk of DNA that we suspect are probably explaining why that chunk is associated with Alzheimer's disease. So it's starting to give us some mechanistic clues about what part of your DNA is contributing to your risk. So we saw all those SNPs on the Manhattan plot, and now what are we going to learn from them as far as biology is concerned? So here is another graph, and these little circles down here, those are going to represent the SNPs, so the regions. And on the y-axis is the effect size. So how strongly does that piece of DNA actually confer risk for Alzheimer's disease? And then on the x-axis is the frequency of that variant. Is it very rare, like we have in these familial forms, or are they common, where 40% of the population has one version of these? Now, one thing I want you to note here is not only are there many different SNPs that confer risk, but when we looked at what the function of those genes are, it turns out they go way beyond just amyloid beta. They actually implicate a number of important processes in the cell and cells that go beyond neurons. So lipid metabolism, the endolysosomal system, which I'll talk a little bit more, immune response, axon development. And so all these different pathways are somehow implicated in the risk and the pathogenesis of Alzheimer's disease. The other thing I wanted to point out is that, I started out this talk talking about familial Alzheimer's disease related to these genes, but really what we're talking about when we talk about common Alzheimer's disease is we're talking about these risk factors. And the idea is that we all have some of these. Some of us may have more of these than others, and that excess burden or that burden of SNPs may be what tips us over the edge into having Alzheimer's disease or not. Now, it's not that simple, but that's the general idea. So, like I said, people can have different amounts. So how do we even estimate that? So we can do what's called a polygenic risk score. And the purpose of this is to get an estimate really of what their risk burden is. And so this is a very simple way to do it. And we have a real geneticist, Dr. Liz Blue, who's on our team, who actually does this for real. But we take the number of SNPs and then we weight it for its effect size, and we can come up with the number. Now, if we do this for a group of control patients, when I say control, I mean healthy, not Alzheimer's disease, we see that there is a normal distribution of that risk score. Some people have very low risk, some people have high, and then a lot of people have something somewhere in the middle. When we plot the risk scores of our Alzheimer's disease patients, say 200 of them, we see they also have a normal distribution of that risk score, but the curve is shifted. There are more people that have a higher burden of risk than the controls do. And that sort of goes along with the hypothesis that these SNPs are somehow conferring risk for Alzheimer's disease. But an important thing to note, I'd say, is that while they are more often high risk, there is a huge overlap between people, between controls and AD as far as their burden of genetic risk, which goes along with this concept that there's a lot of factors that go into presenting with Alzheimer's disease. You know, some of it is pure genetics. A lot of it's probably how you interact with your environment, and then there are probably other systemic and other genetic influences. So the take home is that there are multiple pathways that lead to Alzheimer's disease, and people have different amounts of risk and different types of risk that lead to their Alzheimer's disease. So I mentioned that that this is diverse pathways, diverse cells. So why would that be? Because we're talking about cognition, and that's really just a neuron process. Well, so it turns out that neurons exist in a multicellular environment, and they're constantly communicating, and there are different roles that these cells take. And so there are these cells, these oligodendrocytes. These are the ones that are responsible for myelinating or protecting the nerve processes. The neurons, there's interactions with blood vessels. And then there are also these cells, these microglia. And these are the ones we saw earlier in the talk. And these are going to be the star of the show. So microglia are innate immune cells, and they're resident in the brain. And they're different because they don't just they don't circulate. They actually take up residence in the brain during development, and then they stay there. And they don't, we don't really know how often they replenish themselves, but it's not quick. They do, unlike neurons that generally don't. But they have similar functions to the innate immune cells in the blood. And there are some people that are trying to take advantage of that similarity and think about how can you introduce peripheral immune cells to go and do the job of a microglia when the microglia is involved in genetic diseases. It's a different story, but interesting nonetheless. And then microglia are quite diverse in their behavior. So they, as you can imagine, having such an intimate relationship with neurons, they are part of this protective network to keep neurons healthy and working properly, but they can also be harmful. So I showed the biomarkers, and one of the biomarkers was this red curve, and this is really what we're looking at. So here is a scan of the brain, and so these are the eyes, and the left is healthy, the right is Alzheimer's. And the signal you're seeing is a compound that binds to microglia when they're activated and they're inflammatory. And what you see is that in the healthy brain, there's some amount of inflammatory microglia. But by the time you have Alzheimer's disease, you have a lot more of these inflammatory microglia. So microglia are changing over time. They're changing over the course of disease. And this, like some of the other clues we have, tells us that maybe there's other places that we can intervene. So in our mind, we have more of a holistic view of the drivers of Alzheimer's disease. And not to say that the amyloid hypothesis is wrong, but just that there may be amyloid and tau, but really the brain's reaction to that amyloid and tau is really important in terms of driving Alzheimer's disease. So to the purpose of the talk, how do immune cells contribute to Alzheimer's disease? The simple answer is that inflammatory microglia, when they're inflamed, they release toxic molecules and eventually lead to neuronal death. So inflammation bad, kills the neurons. Well, that seemed like the logical next step would be to let's just try an anti-inflammatory. And that made sense also because epidemiological studies had demonstrated or had shown an association between use of nonsteroidal anti-inflammatories and protection from Alzheimer's disease. So we tried one of these trials with NSAIDs, and it didn't work. And it wasn't clear exactly why. There's a lot of different factors when you do a controlled study. But I think the big take home at that point is that we really had to get more specific about what we were doing when we're trying to manipulate the immune system. We can't just shut them all down. We had to figure out what exactly we were going to shut down or we were going to boost. And this is where the complexity of the microglia come in, because there are not just anti-inflammatory and pro-inflammatory microglia, there are many different kinds. And they all have different jobs, which is really how I'm defining them, is by their job. And so there are ones that are surveillance, and so they sit in the brain and they're moving their processes and they're looking to see if there's any garbage out there or if there's any neuron synapses that need some help. And then there are some that are responding to an infection and they're releasing some cytokines. Then there are kinds that are interacting with the aggregates in the brain, like the amyloid beta. This type of microglia is important, given that part of not only the Alzheimer's disease process, but importantly, part of the mechanism that these new drugs are trying to use, which is to get the immune system to remove amyloid. This type of microglia behavior is really important. So, but we need to establish this. So which of all these microglia are really important? What do we need to pay attention to? And once we figure out what those types are, what is the biology of those cells? Because if we can figure out what's driving those cells, maybe that's where you can intervene. Because the whole point of all this is that we want to know where we can drug. One of the huge problems we've had with Alzheimer's, aside from the ones that I've been talking about, is that it is so difficult to study because you can't just go in and take a piece of brain from a living person, that we don't know a lot about what the mechanism of disease is. And really, we need that to be better about specific and effective therapies. And then lastly, what's driving that inflammation? What's making the microglia inflammatory? It could be the amyloid, but what else is going on? Because if you can identify that, maybe that's something else that you can address. So some of the ways we can try to figure out what the biology is of a cell is to look at the expression of its genes. And sometimes the levels of that expression can indicate if that gene is active, but it gives us a profile, it gives us a signature. And so what we do is we take a brain, and this from here, so some of this data is from our group, and I want to say it's an amazingly communicative and collaborative group. And so I'll give the thank yous later, but Liz Blue, Jessica Young, and then our team members have all been involved in this. So we take a piece of autopsy brain. This is not a living person. We take a little chunk, and then we analyze the gene expression. And so here, each row here is a gene, and it's a gene level. Each column here is a person. And so what you can see is there are some people that have low levels of these genes, maybe high levels of these genes. And we can then take that information and we can examine how those genes interact. We can map them together and we can say, well, not only are these genes different in this person, but also it seems like the overall pathways of X, Y, and Z are also different in these people. And that might explain that biology can explain why there's differences in the cell function. So one of the problems, though, is that if we were to try and take a little piece of brain tissue and examine the microglia, it'll be difficult because all those different types that we're trying to resolve are actually in a very small piece of brain. So here is a really tiny piece of brain. And already in this small set, we see a bunch of different types of microglia, and they look sort of similar. I appreciate that. But so for instance, there's this amoeboid one. So these processes are really thick, they're short, it's more activated. There's this microglia that's one of those degradation microglia that's sitting there interacting with amyloid. So amyloid is in white, by the way, that amyloid peptide. And so you can see it surrounding the amyloid. It's busy doing something. And you also have microglia here with very thin, long processes that are doing more of that motile surveillance kind of activity. So if we really want to get to the biology, the gene expression of those microglia, we need to separate them out. So we took the brain pieces and then we isolate the microglia. We separate them out and then we measure the gene expression for each cell. So now you can say, okay, so all these 10,000 cells, what is the gene expression of each one? So we do that and then we look at that data. And what we find is that they all sort of, the microglia that are similar to each other kind of cluster together. And we end up having 10 different types of microglia. So here is another one of these graphs of genes. What you see is that there are 10 types, and some of these types have really high expression of a set of genes. Another type, say this first one over here, has a really high expression of these genes. And these genes happen to be the surveillance homeostatic ones. So just by the pattern of their gene expression, we can parcel them out. And then we can also then use that information to say, well, what's going on in those cells? So this is the gene expression pattern. What else does it mean? So like I said, this first one had a signature of homeostasis or surveillance. Then there are some that have a signature of senescence. So senescence in immune cells is still a complicated topic and not the same as senescence in cancer cells, and we're still trying to figure out, but nevertheless, these cells have the signature. And then there's some that are more motile. And then there's this cluster here that had an endolysosomal and inflammatory phenotype. What was interesting about this particular cluster is that that is the one that is enriched in Alzheimer's disease compared to control. All of these we see in age donors and AD, but this one in particular was the one that was related to Alzheimer's disease. Now that was particularly interesting that this endolysosomal cluster was enhanced in AD because recall the endolysosomal system is, or you can note, is one of the pathways that were highlighted in our GWAS, in our genome-wide association studies. So we already suspected the endolysosomal system may be involved. And so what does it do? It is responsible for internalizing material from the outside of the cell, trafficking it to where it needs to go, degrading it, I should use this. So it can bring in molecules, it goes into the endosome, it goes into the lysosome, which is very acidic, and it helps to break down proteins and different parts of the organelles, et cetera. And then either it can shoot it out or it can recycle some of it and it goes back up to the top, back to the cell. So this process is extremely important, especially for a cell where the whole goal of the cell is to clear out debris. Now, what's amazing is that these genes that were identified through GWAS that raise the specter of endolysosomal function are all here in this network. And so that really made us think, this hypothesis, well, maybe endolysosomal risk. So maybe people that have endolysosomal risk have problems in their endolysosomal system, and that's why they're getting Alzheimer's disease. And that's a specific kind of Alzheimer's disease for them. So we went back to our cohort of individuals and we generated polygenic risk scores for all of them. And so here is another graph. And this is both healthy donors, so age-matched controls, and then Alzheimer's disease. And this number here is the risk score, and you can see that there's a normal distribution of individuals that don't have Alzheimer's disease. And then there is somewhat of a distribution, it may actually be bimodal, of Alzheimer's disease. But what was notable is that there tend to be more people with high risk in the Alzheimer's disease group. So we said, let's look at the Alzheimer's disease people. Let's look at the people that have high endosomal risk versus low endosomal risk, and maybe we'll learn something about that biology. What distinguishes those two groups? So we took that cohort and we first started out looking at their brain tissue. And what we wanted to do is just look at their microglia alone. Are there microglia and is their endolysosomal system, are they abnormal in any way in the high versus low risk group? And what we found is that there is a small difference. And so this is a microglia cell, it's in green. And then in yellow are the lysosomes. And so here's just a picture of the lysosomes itself. So we can use this immunohistochemistry to label cells, and then we can use machine learning techniques and other applications to get a computer rendering of those lysosomes so it makes them more easy to quantify. And what we found is that the lysosome volume in individuals that had a high risk were somewhat larger than the individuals with low risk. Now, all of these individuals had Alzheimer's disease, so it wasn't just because they had AD. It seemed like there was something possibly related to the genetics itself. And these are all preliminary findings, but I'm trying to sort of give you a sense of how we think about this and how we go investigating it. So what was exciting or interesting is that the genetics seem to predict the cell changes that we were observing. So I said there were large lysosomes. Does that mean anything? Well, we know from different types of diseases, there are lysosomal storage diseases where the problem is that the lysosome isn't functioning right, and those lysosomes get big. There's other examples of lysosomal damage where we see changes and the lysosomes get big. And the idea is that you have, again, this normal endolysosomal system where old mitochondria or amyloid beta or receptors get processed, digested, and then recycled. In the setting of endolysosomal dysfunction, the system cannot degrade as it should. So there's a buildup of material on the outside. There's also buildup of stuff that should be degraded because it's toxic if it isn't. That's still sitting there, and that can cause an injury response. The lysosome itself can be unstable, and that can lead to activation of what we call the inflammasome, which is a collection of proteins that lead to an inflammatory response. So after looking at the gene expression, we said, what are we learning about the pathways? So we went back to the brain. We looked at how the gene expression is different between high and low, and then we looked to see what are the pathways that were different between people that had high risk and low risk. And what we found is that in people that had Alzheimer's disease, if you have a high load of endolysosomal risk, you have increase, so the purple, the dots are indicating where there's an increase. You have an increase in these pathways of the inflammatory response or cytokines or phagocytosis, whereas the people with low risk score had pathways that could be interpreted as being neuroprotective or at least homeostatic, so negative regulation of cell death and neurotrophin signaling. So it suggested to us at the very least that what's happening is that there are gene expression changes that don't seem that they could be attributable to the pathology itself, since everyone has Alzheimer's disease, but seemed like one contribution could really be that the genetic risk itself was making their system more or less susceptible to injury. So in sum, one example of how genetic risk in immune cells can lead to brain injury is what we discussed today, which is that there may be individuals out there that have a higher load of endolysosomal risk, and that leads to dysfunction of their endolysosomal network, their capacity to digest, which then leads to problems in the microglia function, which can cause changes in gene expression, changes in release of inflammatory molecules, which can both cause neuronal injury, as well as perhaps lose the ability to be neurotropic. So all that's fantastic, what does that mean? So the future, the fantastical future, is that we start out with a group of folks. We can use those biomarkers I was talking about, or maybe eventually even these PRSs, I don't think so, but these biomarkers to identify people early. We can try to understand what the pathways are that are driving their specific Alzheimer's disease, and then you can give them the correct drug that addresses their most important risk pathway. So, but that's way in the future. And I don't want to just leave you with the sense that now we're all just sitting ducks waiting for the dementia to happen until the scientists get their act together. There are many things that we can do right now. So this is from the Lancet, one of the biggest medical journals. What they did is they aggregated all this data about what are risk factors for dementia. And to be clear, this is dementia, not Alzheimer's disease. But these factors affect cognition, and really it doesn't really matter what's in the brain. We really care about what's affecting cognition. And so what they found was that in different stages of life, there were different factors that were particularly important. And so, for instance, if we look at late life, already late life, factors such as depression or smoking, social isolation, those can still have an effect on your risk for dementia, even at later life. In midlife, things like managing hypertension, obesity, diabetes, hearing loss, those are all factors that can affect dementia. Those are all things that we can do for ourselves. And then importantly, is education. And this topic is a bit fraught with Alzheimer's disease. The data is kind of all over the place, as you can imagine. It's extremely complicated because it's never just education. It's all in the context of neighborhood and schools and teachers, et cetera. But we know that education in early life can influence risk for dementia. And so maybe that's reason enough that as a society, we try to think about, if we value cognition, what are the changes that we can make that can really give our next generation the best chance. And in some, their estimate is that about 40% of dementia risk is modifiable. Even if you have cognitive impairment, though, there's stuff you can do. So at the Memory Brain Wellness Clinic, which is at Harborview, we have a number of wonderful programs. So one of them is run by Dr. Chris Rhodes, and it's called the ADAPT program. And it's meant to, it's a 3-week course, and it's meant to really reinforce or build these strategies to live well with memory impairment. And so they have this sort of complex session of exercise and support groups and cognitive rehab. And it's really a way to say, you know, I'm not going to make any changes to my biology of cognition, but I can strengthen my resilience. I can strengthen the strong suits I have right now and continue to enjoy my life and enjoy my interactions with others. So that is all I'm going to tell you about today. And the work that I have shown you from our lab or from this amazing team here in my lab, and then these really important collaborators, Jessica Young and Liz Blue. The team is Katie Prater, Lexi Sotelo, Sinath Momday, Akana Reed, and Corbin Johnson, Isa Smith, and Nick Carragas, and all these folks also had data that I showed. So with that, I am open to questions.
Because I can get started. You mentioned PRSs in clinical practice. When do you think this is going to be something where people are going to get their genetic risk for Alzheimer's at birth and then be told that and live with that?
Yeah, my hope is no. So, you know, the polygenic risk scores in reality have been useful to us just to understand the biology. There are so many caveats to these because, as you can imagine, so we talked about how there's variation in the DNA. There's variation across ancestry. And so a gene mutate or a SNP in one group, say white Europeans, has a different risk than the same SNP in a different group like African Americans. So that means that all this data that we have is about polygenic risk is built upon a population that are important, but really not applicable to other groups. So here is, this is just a graph that, this is just a discussion about how many studies there are in different groups. But really, if you were to look at the prediction ability of a certain polygenic risk score that were based upon Europeans, this predictive accuracy is 1. But if you look at different groups, so South Asians, East Asian, and African, you see that the accuracy itself goes down. So the short answer is we're not ready yet. And I'd say that there's so much more to learn about what the implications are to PRS, what the implications are to, you know, we don't want to give the sense that this is all defined, deterministic, because these is all interactions with our environment.
From Zoom, we have Anthony who asks, do you see any changes in the pathways enriched between early onset AD and patients who develop AD later in life?
That is a fantastic question. And I'll tell you, say in December when I find out if my grant gets funded. because that's exactly what I want to know. And that'll be really interesting to give us more clues about, because the thing about studying people with early onset Alzheimer's disease in their brain tissue is that um, they are not changed by the ravages of time. So unfortunately for them, they die early in the 40s or 50s. And so some of the changes that we know are happening just because of age are not so present in their brain, and it gives us a little bit clearer of idea of what the pathology is.
There you go.
Hi, great talk. When talking about Alzheimer's, you hear all the time about how women are disproportionately affected. Did the data you gathered segregate by sex in any way?
That's a very good question. And I should acknowledge that there are some folks out there that don't believe the sex difference, but it seems like there has to be. But anyway, in our data, what we were able to find is that there were gene changes that were driven more by sex than by anything else. So we do see that. And particularly in immune cells, sex can be an important driver of gene expression, and that's an important factor we have to think about.
To what degree today do we have confidence in our models that we have to study, as far as mouse animal models or the organelle or smaller models that you were talking about, that the phenotypes that we read out are truly representative of the complex phenomena that we see in a living human being?
This is a tremendous issue, tremendous problem. And I don't want to take the easy way out, but to some degree, one of the approaches or frames of thought have been that it's the best we can do, which is not a good answer. So there are ways that people are trying to improve the organoids. Say they're actually measuring not just electrical activity, but somehow they're measuring things like memory in the organoid, which yes.
That mean, right?
That's what does that mean? Right, because these organoids that have only 4 cells and no soul. So that has been a confounder with Alzheimer's research, and I think that explains why we took a sort of billion-dollar tangent. Was in large part because we assumed that the readouts of pathology were appropriate for the human, and there's got to be something to be said about the fact that you can't recapitulate Alzheimer's in a mouse. So yeah, that's a really important question, and it's sort of the 800-pound elephant in the room.
Okay, fair. Thank you.
Great talk. You showed some data from patients before they had cognitive symptoms.
Yeah.
What did they come in for, or how did you get that data?
Yeah, that's a really great question. So that data is compiled data mostly taken from individuals that have the genetic form of disease. And so these are folks that we find out, so their father or mother had genetic Alzheimer's disease, and then they knew they were at risk. And so they would get tested. And then we were able to see, okay, well, based upon your mutation, we expect that you're going to start to have cognitive problems in 15 years. And then, but these people get involved in research and they donate their blood and their CSF and their imaging, and that's how we start to map how those biomarkers change. We know less in people that have the sporadic form, mostly because we don't have that type of structured study. But there are beginning to be certain trials that are looking for amyloid, will screen people even if they don't have cognitive impairment. And so those are times that we can get some of those biomarkers on people that don't actually have cognitive impairment.
Hello. I know that recently there's been kind of a shift in focus for neurodegenerative research to the enteric system. I was wondering if that's also gonna be the future for looking at Alzheimer's, and does genetics have a role in that?
Yeah, that's a really terrific question. And, I have another colleague who's up on that list, Dr. Paul Waldmanis, who's been toying and interested in that idea. But the concept being that your biome, your gut biome, can not only influence cognition, but could it also promote propagation of some of these peptides. A lot of this is known more in Parkinson's disease, where there's evidence to suggest that the problematic protein actually starts in the gut and goes up to the brain. And there are individuals who are looking at that, investigators interested in looking at in Alzheimer's disease as well. No, that's just with regards to whether that peptide gets in the brain. You know, the multiome, not the multiome, the gut biome writ large and how it affects the system is definitely an active area of research. And yes, I think that's going to be really important. And we are starting to collect stool in our samples, in our subjects.
I have a question too as well. You talked a lot about the graveyard of therapies that have been tested out. What are you hopeful for in terms of the therapies that are currently in development, and which ones do you think that we should be not spending our time and effort on because you think that those are going to likely fail?
So, you know, I'm not a trialist and not a know-all, so I can't really comment on what I think isn't going to work. But so I do think that there is some interest in trying to use molecules like rapamycin that might help autophagic pathways. The rapamycin may be too broad of an approach, but it is something it's in trials, and I think it could be interesting. We still need to know more about what it's doing in the different cell types, but something like that could be useful. Yeah, and then there is interest in molecules that could be neuroprotective or enhance neuron health. So not necessarily dealing with removing the bad stuff, but at least giving them some ability to withstand injury, because really likely, you know, like AIDS and HIV, we're not necessarily going to we're probably not going to get rid of Alzheimer's disease, but the goal would be, can you just slow it down to the point where you'd have to be 120 in order to get it? That would be good enough.
I have a simple question. So because most of the data so far is based off of European SNPs, do you think there would be any way there might be more of the brain immune cells out there?
That there may be different contributions from different types of people?
Yes, different types of.
Yeah, absolutely. No, I think there's absolutely reason to think that there are, you know, one of these genes that has kind of some risk in the white population actually has a much higher risk in the African American population, which gives us some clues about what, you know, what their immune system may be doing compared to the white population. So I think there's definitely a lot to be learned. So there is definitely a huge push right now, at least conceptually, to try and expand these cohorts so that we can do a better job of collecting everybody. And that's going to take time. But there have been some studies in smaller groups where we clearly see a difference. And I'm sure some of them are in the immune pathway, and we'll learn more from them.
Any other questions? Any last questions from online? Well, thank you very much. This has been wonderful. I really appreciate you joining us this evening. So
All right. Thank you.